I'm Christina Campbell, here reporting for Microbiome Times, and we are lucky enough to be here today with Dr. Joël Doré.
Hello.
He's Research Director at INRA Michaelis Institute and the Scientific Director of Metagenomic. So thank you for being with us.
Thank you.
So the question I have for you today, how can we group people into different categories based on metagenomic analyses of their gut microbiota?
So we have explored now a large number of individuals, healthy individuals and patients, and we now know that indeed there's high inter-individual variability, and there are in a way clusters or groups of individuals that share commonality or similarities in their microbiota that have allowed to describe what we call enterotypes, a division of the normal population into what we can view as ecological settings of the microbiome. We initially described in 2012 3 different enterotypes. It's been refined in a recent publication, and we now have this vision of one of the enterotypes actually splitting in 2 separate entities, and they are connected, as we view it now, with the richness of the microbiota, such that one of the enterotypes will be low richness and the others probably high richness. And they are also beginning to be reported as connected with health in several aspects. The connection with the richness in itself is an indication, but there's also one of the enterotypes which is dominated by the genus Bacteroides and really low richness that is obviously connected with more fragile environments, and so it will be higher prevalence in patients in most or more severe conditions, so to speak. So that means that just profiling the microbiota of a population, general population or patients, will tell you where they belong in terms of gut ecology at the moment you do the profile, and possibly now we will be able to connect that with a higher risk or lower risk of development. So that could mean a lot for nutritionists or for clinicians.
Mm-hmm. And are there any particular diseases that are of interest for using enterotypes to classify certain groups within it?
So what we have seen so far, as I was mentioning, one of the enterotype with particularly low richness of the microbiome, i.e. low diversity, low species differences. In that case, it's connected with, in terms of scientific literature available today, higher risk of development of metabolic disorders or metabolic conditions, diabetes, obesity, and even comorbidities within. And when I talked of low richness, which is not per se focusing on enterotypes, but specifically splitting the population in high and low richness, this has been documented in many conditions, and we know that low richness is associated with a lesser response to treatments, for example. So it's true for dietary intervention in obesity, it's true for cancer immunotherapy, for example, where we know that low richness is associated with a lesser response to the treatment. It's true for most severe conditions and aggravation in the liver pathway towards the most severe outcome, which is essentially cirrhosis and then severe condition where you need to envision transplant of the liver. That are just a few indications where we are convinced that telling the clinician, for example, the status of the microbiota in terms of richness is very important, because it will allow to help the clinician decide in terms of clinical management of the patient.
And can you tell us, why is it important to standardize the sample collection and analysis if we're moving toward metagenomic data being useful in the clinic?
Yeah, so I'm very keen on that. Actually, standardization is really critical. Now, we know that there can be bias that we introduce in the process of generating data. We want to be able to use the data for nutrition or clinical purposes. And well, if we have this accumulation of bias in the process, then what we tell in terms of biological story in the end can be very wrong. So we absolutely want to secure the robustness and the validity of the information we generate. And yeah, the best way to do that is to really have highly standardized processes, and they start from sample collection actually. So just recommending the proper way of collecting the material, of shipping the material to the place where it will be dealt with, and then among the steps we have to deal with is preparation of DNA from stool material, for example, or biopsies, and then sequencing the material for generating sequence data. All those steps are critical. We have done good efforts, I would say, when it comes to full shotgun metagenomic sequencing in the end. So probably the best way to provide material or information to the clinicians today, such that we are in a situation of really writing and providing the guidelines to be able to have a highly robust and reproducible determination at present. So it's a special moment in that respect, actually.
Yeah, I look forward to seeing how this progresses over time. So thank you for joining us today.
You're welcome.
Dr. Joël Doré.